let's talk a little bit about linked genes. So we mentioned this a little bit in the previous chapter. We were talking about how genes that are on the same chromosome and close together on the same chromosome, it's kind of like being in the same car. If you're in the same vehicle, you're probably going to end up at the same destination. So much like genes on chromosomes, if you're on the same chromosome and the chromosomes are being moved around when gametes are being formed, if you're close enough on the same chromosome, you're probably going to end up being in the same, sorted into the same gamete, right? And so as we think about this, each chromosome has hundreds or thousands of genes depending on how long the chromosome is. Only exception to the rule here is the Y chromosome. It doesn't have that many genes on it. But genes that are located on the same chromosome will tend to be inherited together, and they are going to be called linked genes. Now, Thomas Hunt Morgan did a lot of other experiments, genetic experiments with fruit flies to look at gene linkage. I mean, he was able to, before we could do DNA sequencing, he was able to identify the sequence or the order of genes on a fruit fly chromosome just by doing a whole bunch of breedings to see how frequently certain genes or certain characters were passed on to the next generation together. So really cool. Read about it in chapter 15. But he did a lot of work. Thomas Hunt Morgan did a lot of work looking at linked genes. Now, just to give you a little bit of an example here, right, we are looking at two chromosomes, so these are homologs, same length, same shape, meaning the centromere here is in the same position, and it's got the same genes A, B, and C. You'll notice that one chromosome has 3 recessive alleles for these genes, and the other one has 3 dominant alleles. And so if a cell containing this pair of homologs were to go through meiosis, the first thing the cell would do is it would duplicate all of its chromosomes, as you see here. Now we have 2 copies of each homolog. And then the copies would be separated into the 4 daughter cells that would make the gametes, right? So 50% of the daughter cells would get the chromosome copies with the recessive alleles, and 50% would get the chromosome copies with the dominant alleles. Now, remember in the previous chapter, I asked you to think about, well, when we were talking about these linked genes, there was a way in which linked genes can become unlinked, especially if genes are farther apart from each other on the same chromosome, maybe like A and C here. So can you think of something that could happen during meiosis that would unlink the recessive alleles from the other recessive alleles and allow you to maybe transmit a combination of dominant and recessive alleles for these genes to the next generation? I hope you mentioned crossing over, right? And so crossing over happens at prophase 1 of meiosis. We can see it happening right here between this white and blue homolog, or non-sister chromatids in this case. And when you cross over here between the A, B, and the C genes, now you create these recombinant chromosomes that have a little bit of mom's DNA, a little bit of dad's DNA, and you're no longer transmitting only recessive or only dominant alleles, right? Now you have a mix. A, B are recessive with a dominant C, or A, B are dominant with a recessive C allele. And so this is one of the reasons why the Mendel's law, one of them, it doesn't apply to these genes that are far apart on the same chromosome, because you can have now the process of crossing over separating those genes during gamete formation. Okay, so one of the last things then we kind of need to discuss is we've looked at all the normal events that can happen during inheritance. What we didn't get a chance to talk about was what happens when things don't go as planned? What happens when mistakes are made? And so this particular slide introduces one particular occurrence that's called nondisjunction, where chromosomes don't separate properly during gamete formation. And so nondisjunction means that either homologous chromosomes don't separate properly, or sister chromatids of one duplicated chromosome don't separate properly. And what this does is it ends up creating gametes that have either too many or too few copies of a certain chromosome. Let's take a look at what happens when non-disjunction takes place in meiosis 1. This is shown here on the left side of this figure. So if you have in meiosis 1, the homologous pairs lining up at the metaphase plate, one pair up here at the top separates perfectly fine, the other pair suffers a non-disjunction event. That means both homologs are going to be pulled to one of the daughter cells, and the other daughter cell is going to be missing a copy of that particular chromosome. As meiosis 2 then finishes up, all of the gametes that were formed from this meiosis event are going to be wrong, right? They're going to have too much or too little genetic information. 50% of the gametes are going to have an extra chromosome, and 50% are going to be missing a chromosome. This non-disjunction event can also happen in meiosis 2. This is not as severe because 50% of the resulting gametes do come out being normal, whereas the other 50% are abnormal. So let's take a look at non-disjunction meiosis 2 here on the left. So meiosis 1 happens perfectly fine. You pair up the homologs, and then you separate the homologs into individual daughter cells, and then take these daughter cells through one more cell division. The daughter cell on the right goes through proper meiosis 2, no problem, and each daughter cell ends up with one long and one short chromosome here. These are the normal gametes. On the other side, though, this daughter cell suffers a non-disjunction event. So one of the two chromosomes here, the sister chromatids, don't separate properly. And so sister chromatids, both of them are pulled to one side, and the other cell is missing a copy. So in this case, 50% of the total number of gametes that are formed are abnormal. 25% have an extra chromosome, and then 25% are missing a chromosome. Now, if this happens, it's really not a big deal unless the gametes that have the wrong number of chromosomes are actually used in a fertilization event, right? So for example, if you were to take this chromosome right here that has an extra copy of the this cell right here that has an extra copy of the long chromosome and allow it to be fertilized by this cell that's normal, in this case, you would end up with 2 copies of the short chromosome. That's good. That's how many we want. Then we'd end up with 3 copies of the long chromosome. That's too many copies. Or if this cell, for example, was fertilized by this cell, in this case, again, 2 copies of the short chromosome, no problem there, but you're missing a copy of the long chromosome. That's a problem. And so when you have this fertilization event that takes place with gametes where non-disjunction has occurred, the resulting zygote will suffer a condition that's called aneuploidy. Aneuploidy means you have either too many or too few of a particular chromosome. There are 2 flavors, right? You can either be a monosomic zygote. In this case, you only have 1 copy of a particular chromosome, or you can be a trisomic zygote where you have 3 copies of a particular chromosome. Monosomic zygotes are very, very rare in humans. And we'll talk about only one condition where you can have a monosomy, so only one of a particular chromosome, and still survive through gestation and be born and survive through infancy and childhood. And then trisomic zygotes, these are a little bit more common, and one of maybe the most common or well-known trisomies is trisomy 21, which is also Down syndrome. And so this is a karyotype from a Down syndrome patient, and you'll notice that all the chromosomes have the right number, right, 2 of each, except for when you get to the 21st chromosome, and here this individual has 3 copies of that chromosome instead of just the expected 2. And because now you think about it, you're like, well, you're not missing any genetic information. How bad could that possibly be? But it is really bad because remember that your cells, your body is really used to having a normal gene dosage, right? 2 copies of every gene. But when all of a sudden you have 3 copies of all of the genes on this whole chromosome, that's going to mess up a lot of things. And this is why we call it a syndrome, because there are a lot of side effects that come along with having this particular disorder, from mental retardation to specific facial structural features, stature differences. A lot can go wrong. You can also have aneuploidy that takes place with the sex chromosomes, and there's a variety of different combinations that can result when you have aneuploidy with the sex chromosomes. So if you have an individual that inherits 3 X chromosomes, these individuals develop as females, right? There's no Y chromosome, so nothing to take them down the path of developing male sexual characteristics. And typically, XXX females don't have any unusual physical features, and they tend to be fertile, and you wouldn't know just by looking at somebody that they have 3 X's. You can also have 2 X's and a Y. This is called Klinefelter syndrome. This individual will develop as a male because there is a Y chromosome, so the SRY gene is present, and their reproductive tract is pushed down the path of developing male sexual characteristics. And then lastly, we have monosomy X. This is called Turner syndrome, and this is the only known viable monosomy in humans. So really, the only chromosome you can have 1 copy of and still survive is the X chromosome. And these individuals develop as females, but they have other issues that also arise with only having 1 copy of the X chromosome. Okay, so aneuploidy, which we just talked about, is when you have more than 2 or only 1 copy of a certain chromosome. But in some organisms, typically plants, you can actually have multiple sets of the full set of chromosomes. So instead of just having 2, you can have 3 sets or 4 sets or 10 sets even in some cases. And this is called polyploidy. So examples are triploidy, where you have 3 sets of chromosomes, tetraploid, you have 4 sets of chromosomes. This is very common in plants. Many of the produce that you eat, like strawberries and kiwis and watermelons, are actually polyploids. Their nuclei are very crowded with more than just 2 sets of chromosomes. Animals don't show polyploidy. And polyploids are also much more normal in appearance than aneuploids are. This again comes to that gene dosage effect, right? It's a lot worse to have an imbalance in the copy number of genes. So if you have too many of certain genes but fewer of others, that imbalance is a lot worse for the organism than having extra copies of everything and everybody, all the genes kind of being equal. So polyploids are definitely worse. Aneuploids are definitely worse off than polyploids.